Well, thank you everybody for coming. Those of you who know me probably know that I was at the very first SENS conference, and at that point I was not even a student. And now I've actually got a bachelor's degree, and I'm now working on a PhD degree at the tender age of 50 years old. So I appreciate very much, and I'm humbled by the SENS Foundation people who have helped me here and are now supporting the research that I'm doing in Judy Campisi's lab at the Buck Institute. So with that, it's a PhD project, and I wish it was incredibly groundbreaking and novel. Who knows, it could turn out to be. Maybe the end of the story is not over yet. But so I'm getting my degree from the University of Alberta, and I've been at the Buck Institute. And I'm going to talk to you, the outline of my talk is two parts. The first part is the senescence-associated secretory phenotype, which you've probably already heard about, enough about, and its relationship to age-related disease. I'm going to talk to you about a screen that was done in the lab and how a portion of my project was dealing with validating and identifying candidates of that screen that were able to lower components of the SASP. And one of those candidates that came out was a flavonoid, Apigenin, which is a natural product, which is quite good at lowering the SASP components. And then I'm going to just briefly talk about another project which I have ongoing, which is dealing with the Buck Institute Polyketide Library. So inflammaging you've already heard about, but it bears repeating because inflammation is at the center, or pretty much the core of almost every degenerative disease that we experience as we age. However, its origins remain unclear, although senescent cells have been proposed to be a part of that. The senescent phenotype is, again, I'm just going over old ground, is essentially irreversible growth arrest in response to some cellular damage. There is a resistance to apoptosis, as well as altered gene expression, of which the SASP is a component of that as well. Mostly, senescence has been known to occur in response to some sort of genomic or information damage, as you heard from the previous speaker. Chromatin instability plays a role, non-telomeric damage, shortened telomeres with replicative senescence, oncogene-induced senescence, as well as strong mitogenic signals. Even just sometimes FBS, can sometimes lead to rapid senescence in PD, in doublings, population doublings of your fibroblasts. So senescent cells accumulate in vivo. We pretty much know that. They increase with age in all mammals, and they are at all the sites of age-related pathology that we've actually looked at. And the beta-galactosidase stain, which was pioneered in the lab of Judy Campisi, is one of those markers. It's not a perfect marker for senescence at all, but it is one of the gold standards that a lot of people use to determine whether a cell is senescent, whether it's present in a degenerative tissue. And they are present in most degenerative tissues. In fact, emphysema, this is just one example of many. In the lungs of emphysemic individuals, there is about 15% beta-galactosidase positive fibroblasts in an emphysemic lung. Currently, COPD, which is partially caused by emphysema, is an unmet need. And there is the inflammation that's associated with COPD. They don't really have a way of controlling it. They don't know why the drugs that they're using even sometimes work or sometimes don't. And it could be because these are senescent cells which are present in the lung. This is an example of a protein antibody array that was used in the Campisi lab to determine the components of the SASP originally. The top panel is senescent cells, and the bottom panel is non-senescent pre-cells or pre-senescent cells. Interestingly, and most people already know this, but perhaps it bears repeating, we have 3% oxygen and 20% oxygen concentrations. Most experiments in the past were done at 20% oxygen concentrations. And if you notice that yellow is an upregulation of an inflammatory cytokine and blue is a downregulation, you see that at 3%, senescent cells secrete an awful lot more cytokine, pro-inflammatory cytokines than at 20%. So when we're reading the literature, we really have to be careful that we know what incubation and what those conditions are. But as you can see, in all of these different fibroblast strains, both at 3% and 20% pro-inflammatory cytokines are there. In the lab, we use IL-6 as our sentinel marker for the SASP, for the secretion of the SASP. Although you probably can't read those, but there's IL-6, GRO alpha, and IL-8 are at the very top. And so we do look for those when we look at the effects of any intervention on the SASP. Oh, going back is not what I want to do. Okay, so the screen in the lab and all of our screens, it's not a rocket science, but basically we irradiate fibroblasts with 10 grays of X-rays. We cause double-stranded breaks in the DNA. The cell becomes senescent, enlarges in morphology, begins to secrete some of these SASP factors, and we just treat these cells over time with a drug to determine whether or not the level of IL-6 can go down or up. We use a very sensitive assay, which is an antibody-based assay. It can measure in the picograms of substance, and it is just a excitation strategy, and we can see that very small levels of IL-6. The Prestwick library, which was the original screen, which was done in the lab, is 1,400 FDA-approved compounds in clinical use, and 20% were found to lower IL-6 secretion, some of them because they were toxic, some of them because they were not. We did measure viability via ATP. The red arrow points to the compound that I pulled out eventually after testing an awful lot of those candidates, and it's Apigenin. It's a simple flavonoid. There's 5,000 flavonoids, over 5,000 flavonoids. This one is called Apigenin, and it is isolated from various foods. As you can see there, parsley is the one that I'm using. It's from Sigma. It's amazing how expensive parsley is from Sigma. But anyway, so Apigenin was what I focused on, and I did a dose dependency on senescent fibroblasts in both BJs and HCA2s, and we found out that 10 micromolar of Apigenin was sort of the minimum dose that gave us the maximum suppression of IL-6. And so we settled on that, and we went forward to look at IL-6 secretion via immunofluorescence, and we saw that irradiated fibroblasts treated with DMSO do show actually double the number of positive cells for IL-6 than Apigenin. So we do see a suppression of IL-6 in that regard. I don't know if you can see too many of those. That's actually not bad. Of course, the gold standard of beta-galactosidase, you want to see what effects any drug treatment has on your beta-galactosidase staining, and we found that Apigenin treatment did not increase beta-galactosidase staining in non-senescent cells, and we saw that it didn't reduce the beta-galactosidase staining in irradiated cells. So that was comforting. At least we knew that we weren't reversing senescence or causing cells to re-enter the cell cycle, because that's not something you want to do with a senescent cell. You also don't want to cause too much problems for non-senescent cells if you're looking at an in vivo treatment of potential clinical application. So I did a dose concentration-dependent inhibition experiment over 10 days, measuring population doublings. And we did find that at 10 micromolar, you do see a inhibition of growth in non-senescent fibroblasts of about 25%. Day 5 is when I actually released those fibroblasts from treatment just to see whether or not they would re-enter the cell cycle, and they do. So there isn't something happening that's causing some sort of irreversible inhibition by Apigenin. So we were gratified by that. 25% is something we thought we could live with. Again, you don't want to be reversing cellular senescence. You don't want senescent cells to re-enter the cell cycle as a result of your drug treatment, and we found that with EDU labeling, which is a fluorescent probe that's incorporated only into dividing cells, that the differences between DMSO and Apigenin, there was no difference. They were both about the same. So we were happy with that. Another reason why cells can actually stop secreting is because they're dying, and just because they could actually be dead and on the plate undergoing apoptosis and not secreting. So we wanted to test that and make sure that the cells were not secreting because they were dying. We did a caspase assay with luminescence. Staurosporine is a positive control, and you can see that Apigenin at 10 micromolar did not induce either non-senescent or senescent cells to undergo apoptosis, although it has been reported to do so in cancer treatments. So in a clinical application, you kind of want to know what the pharmacodynamics are in terms of treatment. With rapamycin, for instance, if you treat cells for 24 hours, that effect remains for at least up to a week to 10 days. If we were going to use Apigenin as a clinical treatment, would a treatment over 24 hours early be good enough? Would that create a lasting effect? It doesn't appear so. Both long-term treatment or releasing cells from treatment both see that Apigenin needs to be continuously applied in order to see a suppression of the SASP in this 10-day treatment or release experiment that was done. So not only is IL-6 the sentinel, but you want to look at the entire component of the SASP. We could have done the antibody microarrays that were done before, but they are incredibly expensive. They're not that accurate, and you have to do an awful lot of them to get statistical power. There is a new technology out, some of you may have heard of, maybe even used. It's called the Luminex, and at the time, Stanford had a really nice little machine. You could just send them a sample for $85. You would do duplicates, and they'd measure 51 different cytokines. So that was quite nice, and I was particularly gratified to see that the SASP factors, IL-8, Gro alpha, and IL-6, which were known to come out on the antibody array, were still present. So the experiment worked, and I was also happy to see that there was a pretty dramatic reduction, this is a logarithmic scale, in all of those factors by Apigenin. Nicely, there are different factors in the Luminex that did not exist in the antibody array. IP10 is one of them. It's a chemokine. It wasn't known before that it was a SASP factor or that it was reduced by any of these treatments, but it is also, being a chemokine led us to start looking at the character of the other cytokines of the SASP, and a lot of them are chemokines, indicating potentially that the SASP is a recruitment signal to try to bring immune cells into the environment potentially to help a regenerative response. So that may be a direction we're going to start looking at in the future. We put a 30% cutoff, and we wanted to compare the difference between, we want to make general assumptions or general comparisons between different cell lines to make sure that you're not just looking at cell line specific changes. So we did a 30% cutoff, and we saw that, again, Gro alpha, IL-8, IP10, and IL-6 were the top candidates that were reduced, although we did come up with some others. And the IL-1 alpha is one that I think we're going to talk about in a little while, but these are sort of a fingerprint, general fingerprint for at least dermal fibroblasts in terms of the factors of the SASP that are reduced with Apigenin. We wanted to verify that this was happening not just at the protein level, but maybe it was happening at the mRNA transcription level. We weren't just doing something for degradation. So we verified that IL-6, IL-1 alpha, IL-1 beta is another factor. All of those are decreased in their transcription, which led us to think that the suppression by Apigenin is going to be at the transcription factors which control the mRNA production of these factors. So how does it work? I'm not going to talk to you about the painful months that I spent on things that didn't work. Protein aggregation is not the reason. Inhibition of mucin 1, and neither is inhibition of casein kinase 2, all of which are known to be targets for Apigenin, and not the reason why the SASP is decreased. So we continued on, and we looked at the DNA damage pathway. We looked at both RAS overexpression and oncogene-induced senescence, MKK6, which is a hyperproliferation signal, P38, which is the center, which is operated on both of those, and finally we started looking at NF-kappa B, which is one of the transcription factors for IL-6. So in oncogene-induced senescence, we were curious, does Apigenin lower that? If it doesn't, then potentially Apigenin is acting upstream of these factors. But Apigenin does suppress IL-6 in the SASP produced by both MKK6 and RAS-induced oncogene senescence. We also looked at the DNA damage response in terms of immunofluorescence, and we looked at gamma H2AX foci and 53pp1 foci, none of which changed with Apigenin. So the DDR, at least those components of the DDR, were probably not affected by Apigenin. We looked at phosphorylation of p38P as one of the main orchestrators of activating NF-kappa B, and we saw that there was a slight reduction in phosphorylation of p38 in both IMRs and BJs. Not terribly convincing, so we thought we'd look at NF-kappa B activation itself, and we saw that there was a 50% decrease in NF-kappa B activity, which doesn't necessarily explain the fourfold decrease in mRNA transcription of IL-6. So there are other transcription factors of IL-6 that bind to the IL-6 promoter, AP1 and CEBP, and there may be some actual activity there. We thought perhaps NF-kappa B activity was suppressed by IL-1 alpha, inhibiting the IL-1 alpha loop. So here, one of the targets of NF-kappa B is IL-6, but it's also IL-1 alpha. IL-1 alpha will then feed back, engage the IL-1 alpha receptor, and cause, again, activation of NF-kappa B, creating a feedback loop, which ultimately will raise the level of all of the rest of the transcription targets of NF-kappa B. Could be that Apigenin was actually impairing this loop somehow. So we started to look at that, and it's one of the more recent things we've tried. First, we looked at what does IL-1 alpha treatment do to both non-senescent and senescent cells, and we could see that it actually maxed them both out at approximately the same level, which I don't think was known before, which is potentially an interesting thing. This could be the upper threshold of what these cells are capable of producing in terms of IL-6. We looked at, the next step was looking at the application of IL-1 alpha, doing a titration against non-senescent HCA2 fibroblasts, and we do see that Apigenin treatment does lower the secretion of IL-6 stimulated by IL-1 alpha in non-senescent fibroblasts. We have not done this experiment in X-rayed fibroblasts yet, but that is ongoing, and that's going to be the next thing. So the summary, Apigenin reduces many components of the SASP, potentially by interfering with the IL-1 alpha feedback loop. Future work with Apigenin and novel anti-SASP compounds could lead to treatments for conditions such as COPD, where senescent cells are shown to accumulate. Now, in the part 2, there's just a couple slides. We were looking at a library of polyketides. There was 242 compounds. It was a proprietary library. The company provided the rights to the Buck Institute after a while. And I did a screen, and I found out that some HSP90 inhibitors didn't really decrease the ATP viability of the fibroblast, but it really did knock down IL-6. So this is ongoing. We're going to be looking at HSP90 inhibition, and hopefully something may come of that. I did also do a secondary screen on an additional 30-some of targets, and of all of the 150 compounds, some were toxic, some weren't, some knocked down IL-6, but this particular compound caused major autophagy. The cells eventually died by day 4, but at 2 micromolar, the cells, it could be toxic at 2 micromolar. It might induce autophagy at a lower concentration, so I'm pretty excited to try this on both senescent and non-senescent cells. So I really thank you very much for sticking with me. We have identified a number of novel substances able to reduce a SASP, Apigenin and other Prestwick library candidates, as well as polyketiate HSP90 inhibitors. We've characterized the effects of Apigenin and have identified some novel SASP factors like IP10, and potentially identified IL-1 alpha signaling as the mechanism by which Apigenin provides its effects. And we'll be looking into in vivo experiments in the near future with Apigenin, as well as the polyketide library. Thank you very much to Robert Burrell, Judith Campisi, Pierre Dupre, who's in the lab, all of my lab co-workers who have been instrumental in providing me unending guidance and doing page gels and a whole bunch of other things. And a special thanks to Sen's intern, who isn't here, but I'm not sure if he has a poster, but is Sam Curran. He helped me generate some of the data that I showed you today. And thank you very much. I'll take questions.